the sin of doing this type of model. I think the complications I'm going to talk about are not nearly ready for renewal equation, but we'll, of course, when I have a mathematical problem in Utrecht, I also talk to Odo, right? So maybe this will change in the future. And the second confession is that although I asked all speakers in joint sessions to communicate with each other and interact and make sure their talks are really meshed well, we didn't talk at all. So I apologize for that. So it's going to be something completely different. In fact, I'm going to continue more or less where Andy left off on Tuesday and talk about another complication that we have not yet studied enough. And I think we couldn't study this 20 years ago because there simply were no ecological data in this area. But that has changed substantially. And this is just a very small selection. I think a PhD student of mine came up with 50 or 60, maybe even 70 studies that have been done in recent 2 decades. And this is just a very small selection of that to show you that it's all sorts of hosts in all sorts of ecosystems and all sorts of level of influence that these infectious agents have on either the ecosystem level or energy flow or all sorts of levels of biological integration. And also the other way around, that the community, energy flow, biodiversity, or the ecosystem has influence on what various pathogens actually do and how their dynamics develops. So I think we are now in a situation that we really have to address these questions. It's just that the theory in this case is lagging behind. There's lots of data, but there's very little theoretical development and tools and insight yet in systems like this. And in fact, like Andy already said, of course, infectious agents are everywhere. So it would be very hard if you are an ecologist and study a system in the field that you will not be influenced by infectious diseases. It's just that mostly people did not look at these infectious diseases specifically, but they have started to do so recently. And since I don't use the blackboard for this talk like Odo, this gives me the opportunity to show some nice pictures, none of which I made myself. So this is a particularly nice example. I think Andy was involved in this work. I'll show you a few examples of the sort of phenomena that you observe and that are in need of models to try and address them. So this is the observation that tree cover has increased in a number of decades in the Serengeti, and the reason for that could be that there are fewer fires like this that normally take away the tree seedlings, and there are fewer fires because there is less dry grass around, and there is less dry grass because there are more wildebeest. And why are there more wildebeest? Because we eradicated rinderpest. And you see that quite nicely here in this graph. It's really a beautiful paper. You should all read this, I think. So this is rinderpest in the Serengeti. You see it goes down, not because of wildebeest being vaccinated, because of cattle being vaccinated surrounding the Serengeti. And you see the wildebeest population shooting up exponentially and equilibrating at a higher level. So it was really under control by the rinderpest in this situation. I'll come back to this, this chain of events at the end of the talk. Very nice work by somebody actually also in the audience, Spencer Hall. It's a very fascinating system. I'm sure I don't do it justice by just having this one slide. It's experimental work on daphnia, and these daphnia have a predator. And in one case of the study, this predator produces a chemical into the environment which makes daphnia grow to a larger size. And if they're larger, then they filter more water, so they are more susceptible in some way to spores of a fungal parasite. They get more of it, they get more easily infected. And if they're larger, they also produce more of these spores. So they're not only more infectious, more susceptible, but also more infectious. So this type of system is around. I'll skip this one in the interest of time. I'll do this one. This one's nice because it shows another effect, a sort of cascade of events happening in a community. This is a parasite of crickets, and what this parasite does, like several other parasites, it changes behavior of the host, and it makes these hosts go into streams to complete their life cycle, for which they need the fish, and the fish will eat the crickets and then stop eating as much of the benthic community that they usually eat, and these benthic community are then individuals are then there are more of them, so they can eat more of the larvae of the algae, and they can eat more of the leaves. So you have a cascade of events in the ecosystem depending on whether this nematomorph parasite is there or not. So we have to look at in the food web perspective, and then you get pictures like this, where ecologists have tried to make a picture of what the food web looks like. And in Andy's talk, you already saw if you take the normal predator-prey sort of relation, you could include pathogens and parasites simply as species, because of course they are species. So you could have given them a different node and then links to the host species that they infect, and look at all sorts of things. Well, there are some problems with that, as we will see. These are the sort of pictures that Andy already showed. This is one of my favorites. This is a lake food web with lots of fish and some bird species, and these are a number of parasites that they put in with relations to the various hosts they have. This painstaking work, I guess, to get this. Only we shouldn't stare too much at these pictures, because it's unclear how much information you get out of that. And for that, I always like to show this one. I'm not sure everybody knows this one. This is the withdrawal plan for troops from Afghanistan from the US. And then the US Army General said in the New York Times something very insightful about this. Good. So we want to start more basically, right?
Just the known unknowns.
This is just the known unknowns. So there's three ways to think about food webs. Basically, one is to simply only look at the topology, so which species are connected to which species, and you could add, like Andy did, you could add pathogens or parasites as separate species. There's also a way of looking at food webs by looking at the energy flow to the system from lots of energy from the bottom trophic layers to the top trophic layers, and there, of course, here you can distinguish between different levels of biomass, where here they are all the same. And you can look at real interaction webs, so this is interaction strength between different species. It can actually be measured in terms of feeding rates and biomass, etc. And this is the way ecologists can study these food webs. And you could try and include parasites and pathogens in that way. But I think if you think about how to do this, there are two sets of questions. You are interested in the ecology, or you're interested in the epidemiological type questions. And then I think for the modeling, there is a difference, or you could make a difference. And I'm not going through all these questions. You can read them quicker than, and I'm sure you can think of many more or others. But there are questions that you could get away with by not bothering with how prevalent a disease is or how this changes in time. And there are questions, obviously, on the epidemiological side, where you really need to know how many individuals of a given species are infected and how does this change over time. So I'll get back to that. So I'm advocating looking at different approaches. One would be the direct approach that Andy talked about already. So you see the agent as a separate species in this network, and see what this does to the topology and other sorts of questions. Or you have an indirect approach, which is not recognizing them as species, but saying what Odo said, that they are sort of hidden in the host, and you only take their effect into account in splitting the host species in various epidemiological states, like susceptibles, infectious, well, whatever your favorite states are for the infection you're studying. And just a few slides on the direct approach. This is a sort of food web in slightly more detail with energy flow, and you can measure all sorts of things in real systems, and then you can incorporate that into a food web description and look at, for example, stability properties, And one of the things Andy showed was this graph of Bob May, and it has in fact been shown that in this type of setup, the distribution of the strength of interaction between all these species and over the food web really makes that very complex things are nevertheless stable. So it sort of stabilizes things where May said they would be unstable. And it's the distribution that counts. But the question is, we did this a couple of years ago, but the question is, how do infectious agents influence this distribution, this setup, and how would you go about studying that in these systems? It is not entirely clear to me at the moment. I'll skip that. And there are other challenges here in the sort of in this direct approach, because you can make these pictures with new links of a parasite or a pathogen species to a host, but are they really the same links if you're going to analyze this topologically than predator-prey links? Because it is not a normal consumer resource relation, because even if the pathogen or the parasite kills the host, it's still available for food by another predator. So I'm not sure whether we can see them as the same, and how to make this precise before you're going to study effects of path length, complexity, number of trophic levels, all the sorts of things you would do ecologically if you would not bother with the if you would include them as separate species. And much of the current theory on this, including this stability I just showed you, is restricted to systems in steady state. So it would really only be for questions where you could get away with that. So any epidemiological question where you need variation and change in dynamic properties would not be studied in this way. So there's an intermediate view you could take in the sense that suppose you have a predator and a prey, and now instead of just looking at one predator species, I say we have, say it's an SIR type infection, and instead of that you have 3 different interaction strengths with this prey, and this could lead to a different interaction strength overall if you sort of have a weighted average of these interaction strengths, and you could maybe integrate a little bit of this dynamics in this way. Only the problem is that I wouldn't know how to do it because you have to have a submodel somewhere for the changes in the SIR state feeding back into the ecological model. So you see, if you start to think about it, there are all sorts of problems that need to be solved before you can do something like this. In essence, this is like the network models we have seen so far, only here it is not a problem of determining the network. The network is there, and you can it's known and fixed more or less for succession properties, etc. But it's somehow fixed. Only it's the links themselves that have a value, and the value of that changes according to whether the nodes are infected or not, and even the infection itself. The dynamics of that is influenced by the size of these links. So there's a feedback there that makes things complicated. Good. So I'm more for the indirect approach in this case. So I'm going to look at that. Only you can't do all the ecological questions in that way. So there's a trade-off. So basically, what you do in the indirect approach is you say we have a normal ecological model, a predator-prey, like a Lotka-Volterra system or something like that, and you add epidemiological states according to what your disease, what the disease is that you are looking at. And this is possibly the most boring slide I ever made. It is showing you that a lot of people have done this already in mathematical biology community, and these are all papers. Most of these papers, if not all, look at one predator species, one prey species, and various variations of where you put the pathogen, and various variations on how these species grow, and so they all do basically similar things, but it's just one predator and one pathogen, and you want to move away from that because you want to move to systems where you have not only hosts but also non-hosts interacting with each other. And a typical example of a result in this case is that you get these sort of bifurcation these stability plots where you have the feeding rate. So this is a pathogen in a system with a predator and a prey, and the pathogen can infect both the predator and the prey. And then you can see that if the feeding rate is too high for these two, the predator and prey, to coexist, then the pathogen can mediate this coexistence. These are results that have been known for 25 years in math biology. They are well known. I'll come back to that. So, whenever I need to talk about mathematics, I either go to Odo, I always said that, or I go to Mick Roberts. And Mick Roberts is, I don't want to, Odo thank the previous organizers, that's what I want to do as well, because the 6 months I spent here 20 years ago were very, very formative for my later work. And one of the very nice things to come out of it was that I shared an office with Mick Roberts, who I met for the first time. And in the meantime, this I think is 20 years on, is our 20th paper that we wrote together as a result of that first meeting. This incidentally is a paper. It's in an issue of Journal of Mathematical Biosciences, or Journal of Mathematical Biology, that is devoted to Odo's 65th birthday. So what we did was try to look at this in more generality and try to see if you can derive some general properties, because we are in need of a theory rather than looking at special cases of one predator and one prey. We want to include both hosts and non-hosts and want to have some theory. So what we initially did was the easiest thing, which is basically answer Andy's question from Tuesday: is there a way to do next generation matrices for this sort of system? And yes, there is, and it has been done, and it works quite nicely. So this is, I'm not going to go into all the details, but this is a basic ecological model. There's growth of species, there's death of species, can all be density dependent as far as I'm concerned. There's you are lost because you are eaten by a species of type J, and you gain biomass because you eat species of type K with some energy conversion efficiency. You know, if you're an ecologist, you know the type of model. And what basically happens is you of course you write down a Jacobian matrix, which is usually called the community matrix C, which is given here. Details are not important, and then you can study stability of this of the steady state solutions. Now, what we then do is we add, in this case, a simple SI framework. You can do all the things you would want to do for yourself, but this is the simplest thing we could write down. So you have within host species transmission. If you're not host, then you choose the parameters in such a way that you have no transmission going on, etc. So there are host and non-host possible in this system, and you can lose infected individuals because they are eaten by others, and you can gain also infected individuals because you eat someone, a prey that's infected, and if you are both the host, then the infection can pass to you, and you can maybe also get something from environmental transmission, environmental stuff going on. The details are not important, but what's nice is that if you look at the Jacobian matrix and you order the states in a certain way, that you first do all the population sizes and then you do all the sizes of the infected states, and then you get a very nice form of the matrix out, where the constituting matrices have an actual interpretation. So C is the community matrix we had before, so that determines ecological stability in the system. And H is what we call the epidemiological matrix, because it gathers all the information on infected states, creating new infected states. And that, as I will show in a minute, is related to the next generation matrix that Mick talked about. And then D is a matrix that gives you the influence of changes in the ecology of individuals, so how they behave ecologically speaking, because of the fact that they happen to be infected. So maybe infected individuals have a different birth rate or different feeding behavior, changes that are ecologically relevant, but are caused by the fact that by the epidemiology, by the fact that this individual happens to be infected. And B is the other way around, so it's changes in the epidemiology of individuals due to things that go on ecologically speaking. So, for example, density dependence because of the total population size, density dependence acting on infected individuals, which might be different from uninfected individuals. Now, if we look at the infection-free steady state, a lot becomes simpler. So the invasion problem for a disease, so you have an ecological system in a steady state and you throw in an infectious disease, what happens? That's the question. And in that case, this matrix B is 0. And you get the nice thing that the stability problem of this whole system decouples into ecological stability and epidemiological stability. So something determined by C only and something determined by this matrix H. And this matrix H can be written as Mick explained, as transmission plus transition between all these states, and the next generation matrix that we call of large domain, in this case, is given by this expression. And R0 is the spectral radius of that thing. So just so you can write it down for the general model. And this is just one slide to show what this K looks like. This is for the case where you had a predator. I showed this stability plot before, for a predator and a prey with an infection in both. And you see that the R0 here depends on the feeding rate. So you have a trade-off between feeding rate and R0. So just one example of this in a very simple system, and that is that we looked for a bit at the wildebeest grass rinderpest system that I talked about before, just as something you could do with this. So N1 is now grass that's eaten by wildebeest, and the wildebeest can be infected, and they can transmit the infections among themselves. This is a very simplest setup, and you get a matrix like this, where of course now, because we have only one infected state, a wildebeest, in this model, the H is a 1-by-1 matrix, so R0 is simply this, and the epidemiological stability in this case does not depend on the feeding rate. And you get pictures like this, and only look at this one for convenience. So suppose you have a situation where R0 is way above 1, then what this means is that you have a very high value for the grass. You have lots of grass, close to carrying capacity maybe, and you have a very low population of wildebeest feeding on that. And then if you move by some force R0 to below 1, then something changes at 1, and that is that the grass equilibrates at a very low level and the wildebeest equilibrate at a very high level, which of course is exactly what happened in the wildebeest grass ecosystem. I'll skip the afterthought. That's for people interested in sleeping sickness. So, but also here there's so what are you going to do with things like this? This is the only thing we did, and it needs more. It needs more theoretical development. We have not looked at all at non-trivial steady states. So if you have endemic steady states or any other of the steady states in this system, where typically this B matrix is not equal to 0, well, maybe it's simply doing the right sort of linear algebra to get nice relations out. We'll have to see. Of course, what happens in periodic environments that you have a lot. I'm not sure. There, of course, R0 next generation matrix results have been derived for periodic environments. We have to see how this works. I would be interested in doing something else and saying, well, what if we happen if we add one more host species or take away a host species? This would be a way of looking at this what's called the dilution effect, where Pete, I think, is going to talk about. I'm not a big believer in the dilution effect, and certainly not in that it works always in the direction one says it works. But you could say, well, let's add one host and see how that perturbs the system, and whether this indeed goes in a positive or a negative direction with respect to prevalence of a certain disease. And well, these matrices C, B, these different matrices, they could be explored in different ways. Although I don't know how at the moment, but you could look at indicators maybe related to these, certainly these B and D matrices that have the ecological influence on the epidemiology and the epidemiological influence on the ecology, because there might be some interesting information to get out of that and insight. So, and finally, Andy had a question and had a conjecture even on overheads from 20 years ago. I think how this works with parasites with life stages, and could we take this to address what the influence is of these parasites and life stages on stability of the whole system? And I think we can, because we can also, of course, do this next generation thing with parasites with life stages. And we'll try, and we hadn't thought of it before, but we will try and work on that with Andy in the coming weeks that we're here. And as a summary, the only thing I want to say is that if you make a list again of what you want to add, like age and heterogeneity and all those sorts of things, also think about multiple species and community dynamics, please, in the future. Okay, thanks.
Hans, if I understand it correctly, if you split your matrices in the community and the epidemiological matrix, and then you say it's stability, but it's stability of the state where the diseases are not present, is that correct?
It's stability of the infection-free steady state.
But then what have you got? Because I would imagine in an ecosystem what really matters is the stuff, there's everything out there and all these diseases going. What you really would like to know is how that system functions with all the diseases present. I mean, the fact I can get in there, I know because they...
Sure. As I said, this is the first simple, easy step of the whole... I mean, this is something you could do easily, which is what we did. But surely, yeah, the real questions are more complicated.
But how likely is it that this approach will give you insight or easy insight in this? Because of course you can make a model with everything in there. That's not hard. But
no, I have no idea. So maybe that's a so that's why I say there's a direct and an indirect approach. I'm not saying that this is the approach that everybody should take to do these things. It's just that it works in this case. But maybe for that you need more the direct approach is more interesting to look at include the agent as a separate species and see what you can do with that. I mean, it's not an easy problem, so there are no answers. It's just that we have to. The point of the whole thing is to start thinking about it. That's the.
You said at the start, Hans, that there's more data, and that's undoubtedly true. But I'm guessing that these approaches will be very data hungry indeed. So how will you parameterize these models? How will you feed information into these models? Is there going to be enough out there to get useful results?
Well, that's a good question, but so the first thing I mean by having all these data is not specifically that you would have a data set that you could fit these models to, but at least that people have been studying in many different studies the different phenomena that arise that you could wish to study, right? Wish to model. And I know there are some people, like Spencer has made models where he parameterized things with his experiments, so it is possible. But of course, many of these things are very data hungry. Right? The least data hungry one is the direct approach where you simply look at the topology of the whole thing and you include them as species, because you don't have to worry about what is the energy flow, what is the nature, what is the interaction strength. If you go to these questions, they are much more data hungry than the others. But at least we have now phenomena that you can start to think about it. That's the whole point.
So I mean, there are also a network of people who have a website called Webs on the Web, who are putting together the huge sets of data for some of these food web problems, which range from the stuff I mentioned in California, where we've dissected salt marshes to measure the abundance, biomass of everything, free living and parasitic, as much as we can along the coast of California, to lots of people who put topographic webs together, through to things like Serengeti, where there are really detailed, I mean, some of the animals have got individual names, Leo, this, that, and the other, right through to things that whole groups of species we know nothing about. We've had one person work on the insects of the Serengeti in the last 50 years. So there's not only data hungry, but there's huge inequalities in the quality of the data.
Okay, thanks.
Okay, I think the need is next for coffee to turn into more talks. Thank you very much for both of you.